All right, David, you are good to start whenever you wish.
Great. Welcome and thank you for having me. I'm super excited to be talking about my work today on just one single gene in the entire human genome. But I promise I hope you will find it interesting. My name is David Peede. I'm a fifth-year PhD student at Brown University working with Emilio Puerta Sanchez, and I'm all about introgression. And so to start, I just want to reflect on our own species. And despite the rich historical debate on the origin of our own species, today it is widely accepted that autonomically modern humans originated in Africa, followed by a series of migration events that led our ancestors to populate the globe and give rise to the human populations of today. But throughout these historical migration events, modern humans encountered a myriad of different environments and pathogens that our ancestors needed to adapt to. However, when modern humans arrived in Eurasia, they were unexpectedly met by our evolutionary cousins, Neanderthals and Denisovans. While these archaic humans are now extinct, this population overlap provided the opportunity for modern and archaic humans to interbreed. This initial gene flow event, followed by subsequent backcrossing, results in segments of archaic DNA incorporated into the modern human genomes, a phenomenon known as introgression. However, archaic alleles tend to be deleterious on the modern human genetic background, and over many generations, recombination and negative natural selection will purge the majority of this introgressed ancestry. However, work pioneered by Nobel laureate Svante Pääbo now makes it possible to actually sequence DNA from these archaic human remains. And while this ancient DNA is often very fragmented, degraded, and contaminated with modern human DNA, remarkably, it is possible to recover high coverage archaic genomes, for which I'm showing you on the slide the information for the four high coverage genomes we have currently available, which are 3 high coverage Neanderthal, consisting of 1 older Altai Neanderthal and 2 late Neanderthals, the Chagaskara and Vindia, as well as 1 Denisovan genome. And so studying these archaic genomes has revealed that most non-African genomes carry between 1 to 2% Neanderthal ancestry, while Asian and Oceanian genomes carry another 1% of Denisovan ancestry. The majority of this introgressed ancestry found in present-day populations is assumed to be putatively neutral. However, introgressed ancestry may actually provide a fitness advantage on a specific genetic background or environmental context, a phenomenon known as adaptive introgression. One of the hallmark examples of adaptive introgression comes from my advisor's postdoctoral work, where she demonstrated that a Denisovan-like haplotype found in the EPAS1 gene provided the raw genetic material necessary for Tibetans to adapt to life at high altitudes. Follow-up work showed that while the Denisovan introgression event in Tibetans likely occurred roughly 43,000 years ago, positive selection didn't favor this introgressed haplotype for another 30,000 years. Realizing that these alleles need not be adaptive in the archaic population was absolutely mind-blowing to me, as it highlighted that introgression is an important source of genetic variation for natural selection to act on to help modern human populations adapt to new environments. This made me wonder how introgressed archaic variation may have been helpful for other human populations to adapt to new environments, just like Tibetans. And so to me, studying the effects of archaic introgression in populations from the Americas seemed like the natural choice, as these populations have rapidly expanded from Alaska to Chile and encountered a myriad of diverse environments and pathogens along the way. The challenge is that present-day populations from the Americas are recently unmixed due to European colonization and the transatlantic slave trade, necessitating the need to account for different ancestry components in these populations. So I got interested on MUC19 based off this 2019 paper by Reynolds et al., which showed variants in MUC19 being one of the top candidates for positive selection in the indigenous American populations. I got excited after reading that paper as from my advisor's previous work, I had already knew that the Mexicans in the 1000 Genomes Project exhibited a high density of archaic alleles. And so this has led me to the 3 research questions I will present today. The first is MUC19 introgressed? The second is, does MUC19 exhibit signatures of positive selection in the Americas? And lastly, who is a donor? So let's jump right in. So what we did is I used a reference-free method, which just means we can call archaic or introgress tracks without relying on a donor, on having a donor genome. And we're doing this because 3 Neanderthal genomes or 1 Denisovan genomes surely does not represent the total amount of genetic variation in these populations. And so what I did is I just asked a simple question. Does an introgress track overlap MUC19 by any base pair? And so at that site, I then just going to count the number of tracks that cover that position. And in doing so, you get something like this. And what I want you to take away from this is that in this bluish green denoted by AMR, I'm showing you the admixed American populations, Southeast Asians in pink, East Asians in blue, and Europeans in vermilions. And by far, we have statistically more admixed Americans, or put another way, an overrepresentation of introgress tracks in admixed American populations in this overlapping this gene MUC19. So then to focus our analysis on a specific region, I just asked what's the densest contiguous region larger than 40 kb, larger than 40 kb, so that I can be confident that the results that we are seeing are not due to incomplete lineage sorting. And so that results in this 72 kb window that is covered by the most introgress tracks and partially overlaps MUC19, which leads me to the observation that MUC19 is indeed introgressed. So the next question is does it exhibit signatures of positive selection? So the population with the most amount of introgress tracks at MUC19 is Mexicans from Los Angeles in the 1000 Genomes Project. And under a scenario of adaptive regression, we would expect that these introgress tracks are very long and uniform. And so on the x-axis, I'm just going to show you the coordinates for the longest introgress tracks, but I'm going to show you now all the introgress tracks for Mexicans in bluish green, Han Chinese in blue, and Europeans in orange. And what I want you to notice is that for the Mexicans, these tracks are extremely long. They average 333 kb and are uniform. And so then we wanted to use this population branch statistic, which is essentially asking the question, what is the branch length leading to the Mexican population since it's diverged from the Han Chinese and Europeans? And the idea is that long branch lengths are indicative of positive selection. And so notably, in this small 72 kb region, we have 135 Denisovan specific SNPs that are absent in Africa, present in only the Denisovan, not the Neanderthals, and segregating in Mexicans. And all 135 of these SNPs are above this 95 percentile. In contrast, in that 72 kb region, the gray shaded region, we only have 4 Neanderthal SNPs, where the majority of these other Neanderthal SNPs fall out of this region. And of course, we have some human and shared hominin SNPs between the archaics. And what I want you to take away from this is that here we just use an empirical threshold to identify these SNPs under showing signs of positive selections, but we went back and we validated these results using simulations in SLIM, using the latest up-to-date model for Mexican individuals, and can confidently say that we can reject a null model of neutrality or of heterosis. And so at this point in the talk, you're probably thinking to yourself, so Dave, what does MUC19 do? And so that's the million-dollar question. MUC19 is one of the biggest proteins. It generates one of the biggest proteins in the entire human genome, over 8,000 amino acid residues. And as such, it's extremely hard to study from a molecular standpoint. But what I can tell you is that these mutants are characterized by this exonic tandem repeat. In MUC19, we have a 30 base pair motif. And so here on the x-axis, I'm going to show you the archaics, then African populations, Southeast Asians, East Asians, and Europeans, and then lastly, admixed Americans. And what I'm going to show you is the number of these 30 base pair repeat copies. And so if we look at the archaics, none of them have an exceptionally large amount. And I should note this dashed line represents 95 percentile of this copy number distribution. And then if we look at individuals in blue who have no introgress tracks overlapping this region, we see that they tend to have a relatively low amount of repeat copies, especially when we consider individuals who have at least one introgress track in yellow or two introgress tracks in green overlapping this repeat region. And so when we test this, we see an abundance of repeat copies, specifically in admixed American individuals who have at least one introgress track overlapping the repeat region. But we can go a bit further. So if we zoom into MX Mexican specifically, we can better quantify this relationship and see that there is a strong positive relationship between the number of tracks overlapping a repeat region and the number of repeat copies. And similarly, to a lesser extent, we can also do this for the Indigenous American ancestry component in this region. But what I really want to highlight is that when we go back and we use long read sequences, we reveal a massive expansion on the level of a whole exon duplication. And this is quite phenomenal because it's only found in admixed American individuals, and these admixed American individuals carry the exact same Denisovan SNPs that we see at this 72 kb region. So does MUC19 exhibit signatures of positive selection in the Americas? Yes. Okay, so the donor. So what we did is we computed the sequence divergence between African and admixed American haplotypes and the Denisovan, because we thought it was a Denisovan at first. When we look at African haplotypes, we see that all of these haplotypes are very distant from the Denisovan, which makes sense. That's what we'd expect. We don't expect Denisovan introgression in Africa. And when we look at the admixed Americans, we see the canonical bimodal distribution you would expect, where these haplotypes that are close are representing the introgress haplotypes. However, these introgress haplotypes actually aren't significantly closer, which led us to look at these genome-wide patterns of heterozygosity, because we noticed that these two late Neanderthals are at intermediate distances with respect to the Altai. And just to orient you, essentially we'd expect that Africans have the most amount of diversity or a heterozygous site as quantified by heterozygous sites. And these archaics are super inbred and had low population sizes, so we would expect very few. So again, this is similar to the copy number plot, except this time I'm going to show you heterozygous sites. And so when we look at the Denisovan and the Altai Neanderthal, we see what we expect, a low number. But when we look at the Chagaskara and Vindhya, they have an extreme amount. In fact, they surpass any African individual. And if we plot the individuals with one introgress track or two, we see that the individuals with one introgress track have elevated levels of heterozygosity, which made us think that these late Neanderthals also harbor an introgressed haplotype at this region from the Denisovans. And so what we did is we did a two-pronged phasing approach, combining read-based and statistical phasing. And the crux is we can leverage the fact that we have so many introgress haplotypes, and we can actually phase these late Neanderthals. And when we phase these late Neanderthals, we find that they're actually the closest to this introgress haplotype in Mexicans, which we can confirm with our sequence divergence results. For the sake of time, to answer our last question, yes. And to summarize, what we're proposing has happened at this region is that there was a haplotype that originated in a Denisovan population not closely related to the sequence Denisovan, introgression event from Denisovans into late Neanderthals, and then late Neanderthals into admixed American, non-Africans that was positively selected for in admixed Americans. And I would just like to thank my co-author, my funding, my lab. I'm out of time.
So you won't be able to answer any questions verbally, but we do have the option to put questions in the Q&A feature. So you'll be able to answer some questions there. Meanwhile, we're going to move on to our next speaker. So Josh, do you want to share your screen? And you'll probably also want to unmute yourself.
There we go.
And
Sorry about that.
Yep, you can begin whenever you are ready.
All right. Thanks for coming to my talk, everybody. It's been really cool seeing all these Hamilton talks. I'm Josh Knecht. I'm a PhD candidate at Binghamton University in Jay Sobel's lab. I'm really interested in how selection on hybrid seedling traits might cause reproductive isolation. So, real quick reminder on how we measure reproductive isolation. Um, Anything that keeps the gametes from coming together, we call prezygotic isolation. And if you have successful hybrids formed, anything that causes those hybrids to have lower fitness than the parents causes postzygotic isolation, which is what I'm going to be talking about today. Low hybrid fitness. And so in seed plants, we have this really great meta-analysis that was done by Christie et al. Measuring reproductive isolation. And they found that on average, prezygotic isolation on the left-hand side of this figure was stronger than postzygotic isolation. I'm going to argue in this talk that we might be underestimating postzygotic isolation. The primary reason is because we tend to look at fitness in F1s primarily, and that makes sense because they're the first hybrid, and if there's low fitness in the F1s, there's strong reproductive isolation. But that doesn't necessarily mean if there's no fitness cost in F1s that there's no reproductive isolation, because we only test certain genotypes in the F1, and there's the potential for heterosis covering up hybrid incompatibilities. We also tend to look at fitness in the adults often. We measure things like height or biomass as proxies for fitness. So I'm going to try to really focus on seedlings in this talk. And the reason is if you look at this plant in the lower right-hand corner, this particular species can produce dozens of flowers on a fairly small plant. Each flower can produce hundreds of seeds, and they can live for multiple years. But if they're maintaining a stable population, on average, there's only one individual seed that grows up to become an adult to replace its parent. And so there's really strong selection on these seedlings. And so any hybrid incompatibilities that affect these seedlings might be magnified in reproductive isolation. And so I'm looking at a pair of sister lineages in this Mimulus aurantiacus species complex, which is distributed across southern and central California. These two are Calasinus and Longiflorus. Calasinus is in the purple dots here, in the southern Sierra and the eastern transverse range, while Longiflorus is in the western transverse range, kind of around Los Angeles. So you can see they do have some floral differences. The truth is, we don't actually know a ton about what's causing reproductive isolation in this group or how strong it is. My lab mate, Rachel Kruger, is looking at the pollination syndrome. We don't know a ton about it. I'm trying to focus on the postzygotic aspect. So I did this experiment where I made parental crosses, F1 and F2 crosses, and then crossed the F1s with the parents to make bat crosses. I measured seed size and germination timing. I measured uh, leaf area at multiple time points to try to get growth rates. And then after they were growing out for a couple weeks, I stopped watering them to stress them out and went to about 50% mortality and then watered them again to see who survived. And then I did some quantitative genetic analysis on the phenotypes, which we'll talk about in a minute. But to start off with the main results, to get oriented to this figure, I'm going to use similar ones throughout the presentation. The x-axis here is the proportion longiflorus, which is just the proportion of the genome of a cross that comes from the longiflorus parent. The y-axis is going to be the trait of interest. In this case, we're looking at survival in this drought treatment. And then for these crosses, you can see a thick bar, which represents the 50% credible interval, and a thin bar, which represents the 95% credible interval. So right off the bat, we see that there's really no difference in survival probability in the parents in this drought trial. But if we add the F1, it's also in the same space, similar survival as the parents. So if we stopped here, we would conclude that there's no reproductive isolation caused by these seedling traits. But if we look at the recombinant hybrids, we see that the F2 and the bat crosses are depressed below the parents. So indicating that, yes, there is potentially reproductive isolation here. And so thinking about this has confused me a lot. What does it mean when we have no reproductive isolation in F1s, but in the recombinants we do? And so I've really tried to start thinking of it as in a fitness landscape perspective, using this two-dimensional space. So on the x-axis here, we have ancestry proportion. That's just the probability that a randomly drawn allele, it comes from parent 2. The y-axis is ancestry heterozygosity. That's the probability that at a locus, one allele comes from parent 1, the other allele comes from parent 2. And so all possible hybrids are defined in this triangular space. Parent 1 defines the bottom left, parent 2 defines the bottom right, and the F1 defines the upper corner. The F2 ends up smack dab in the middle, and these back crosses are on these upper edges. And this is really cool because if we have phenotypic means for these 6 crosses, we can estimate the 2 locus cumulative effects for phenotypes, and then go on and plot them as fitness landscapes. And so just to build some intuition about how this works, I'm going to use the Dobzhansky-Miller model as a heuristic to think about it. So just as a reminder, in the Dobzhansky-Miller model, we have the ancestral population up in this upper left-hand corner. Species 1 evolves along the left-hand side. Species 2 evolves on the upper side. And these 4 genotypes in the bottom right have been never tested by selection and are potentially incompatible. And so for this particular example, I'm just saying that the bottom right genotype is completely lethal. Everything else has 100% survival. And if we look at the fitness landscape for this, we see that there's a ridge of high fitness going from parent to F1 to the other parent. So the F1s are able to mate with parents and produce high fitness offspring. If we contrast that with this sort of genetic architecture where we have less strong incompatibilities, but with a different architecture, and plot it on this fitness landscape, we get this kind of interesting target shape where we have peaks of high fitness at the parents and the F1. And so even though the F1 has high fitness, if it mates with a parent, you get a back cross, which has low fitness. If it mates with another F1, you have an F2, which has low fitness. So I'm going to argue that if you have this fitness, this valley between the F1s and the parents, that's indicative of reproductive isolation. And then even better if there's a separation between the parents as well. Okay, so we have a genetic model, and we see these interesting differences in survival. What are the phenotypes that are involved with that? So doing a logistic regression with size as a predictor, you see that size is very strongly predictive of survival in this drought trial. So I want to look at phenotypes that cause seedlings to be bigger. And sorry, just jumped ahead of myself here. So we actually do see bigger seedlings for the parents and the F1s on the left-hand side here. So Calcaneus, longiflorus, and F1. They're visibly bigger than the F2s and the back crosses on the right. So what's causing this difference? So during this early stage, they're growing according to an exponential growth model. So we want to look at the size at time 0, the growth rate, and the age at the time of the drought. And so size at time 0 is a function of seed size. The bigger the seed is, the larger the size is going to be on average at time 0. And so if we look at these seed sizes, we see, first of all, it's really interesting. This is the only of these 3 traits that I've looked at where there's a divergence between the parents, where longiflorus is significantly lower than Calcaneus. But the F1 is at least as high as Calcaneus. But again, we see the F2 and back crosses down low. And sorry, I should note that for all these figures with my traits, the y-axis is the phenotype, which is in standard deviations from the mean. Okay, so if we estimate the genetic effects and project it onto this triangular space, we see this kind of tilted target where we have high fitness in the F1 and Calcaneus, a peak also a little bit lower at longiflorus, but we do have this valley of low fitness between the F1 and the parents, and the valley between the parents, so indicating reproductive isolation. If we look at relative growth rate next, as relative growth rate goes up, we would expect size at the start of drought to go up as well. So positive correlation here. And so we see, again, Calcaneus and longiflorus are in kind of the same space, and the F1 as well. Interestingly, here we see the F2 is occupying this same space, but the back crosses are low. If we estimate the genetic effects and put them on this triangle, we see a valley of low fitness between the F1s and the parents, but we see high fitness between the parents. So that's really interesting, and I'd love to get people's thoughts on that. All right, finally, we have age at the start of the drought, and that's determined by how long it takes to germinate. So we expect time to germination to be negatively correlated with size at the start of the drought. And we see, again, Calcaneus, F1, and the Longiflorus all have the fittest values at low time to germinations, and the F2 and the back crosses have longer time to germination. If we put that on this triangular space, remembering that high values here are low for fitness, we have a fitness valley between the F1s and the parents, and then again, the parents are separated from each other. So if we take these estimates and then try to predict size at the start of the drought trial, this is what we see. So we have this really clear valley between the F1s and the parents, but this whole low heterozygosity region seems to have high fitness. So that's a really interesting pattern. Okay, so the conclusions here are that I think these early seedling stages and early life stages in all organisms are probably really important, and small changes in phenotypes might lead to measurable outcomes for reproductive isolation. And then, in terms of thinking about reproductive isolation, just because we don't see any evidence of reproductive isolation in F1s doesn't necessarily mean that there's no reproductive isolation. We need to have a better understanding of the genetic architecture of the phenotypes we're interested in to try to see what's going to happen in the long run. And so with that, I'd like to thank my lab, my advisor, Jay Sobel, Rachel Kruger, and Irene Martinez. My lab mates have been amazing to talk to, Lauren Campbell, the tech, and then the undergrads, the NSF for funding, and then the Society for the Study of Evolution has been really important in my development as a scientist so far. So thank you all for that.
All right, we have plenty of time for questions. So either people can raise their hands and we can have you do it verbally, or you can post something in the question and answer. It's your choice. All right, Catherine, go ahead. You should be able to unmute yourself.
Yes. I may have missed this. Apologies. My screen froze for a second, but did you say whether you did the cross in both directions? Do you see asymmetries in the F1s?
I did not. I wish that I had, but no. Yeah, when we started this, the plans were different than what they ended up. So no, I don't. And I really regret it because I would be interested in seeing like cytonuclear, if there's any cytonuclear type interactions going on. Yeah.
We've all been there.
Yeah.
So in addition to like wishing you had done the crosses in the other direction, anything else you would have liked to have changed with the benefit of hindsight?
Yeah. So like seed size in particular, but all these traits that are really close to the seed are probably affected by maternal effects. And my crossing design can't really tease the maternal effects out. Like I think I have multiple mothers that they have different seed sizes depending on the cross. Um, but yeah, I really wish I had more mothers to try to tease out the maternal effects.
All right, if we have no other questions at the moment, we're going to switch over to our next speaker. If you have another question you are still in the process of formulating, I recommend you put it into the Q&A and we can get things addressed back there. So our next speaker is going to be Devon. Devon, do you want to try sharing your screen? And then you've still got like 30 to 40 seconds before we actually have you start, so you can spend some time getting this all up and running in whatever format is best for you. You will also want to remember to unmute yourself.
Great. Thank you, Mike.
No problem. It is very easy to lose track of that sort of thing.
You're seeing my slides and everything?
Yep, we've got things on your title slide at the moment.
Okay, great.
Obviously, we can hear you, so you should feel free to begin whenever you are comfortable.
Great. Thank you. Yes, so my name is Devon DeRaad, and I did my PhD at the University of Kansas, and I'm now a postdoc at UCLA. And I'm really excited to be here talking to you today about how new species are formed on islands. And the reason I focused my PhD research on islands is that islands are global biodiversity hotspots. And because I wanted to understand the evolutionary mechanisms generating global patterns of biodiversity, I focused on understanding how species accumulate through time in these island hotspots. And this led to a dissertation chapter revealing that the allopatric speciation among kingfishers in the Pacific Islands was so rapid that the mitochondrial genome couldn't keep up, and the deep coalescence of the mitochondrial genome created strong signals of mitonuclear discordance. And a chapter demonstrating how shifting selection pressures through time can explain the paradox of the great speciators, which are interesting avian groups that tend to evolve into distinct lineages on nearly every island that they reach. But throughout my dissertation, I found myself repeatedly coming back to this deceptively simple yet extremely fundamental question: how are new species formed on islands? And of course, I'm not the first person to be interested in understanding how new species formed on islands, and in fact, I would argue this has been a major question since Darwin's voyage to the Galapagos. And later in the 20th century, Meyer and Diamond published really foundational work on this topic, looking at bird diversity in the islands of Northern Melanesia. And across all of this foundational research, there emerged a strong consensus that oceanic islands, despite being these hotspots of global biodiversity, were simply too small to generate sympatric speciation. And Meyer and Diamond really formalized this notion that allopatric speciation is the rule among vertebrate species found on islands by applying Wilson's taxon cycle hypothesis to the birds of Northern Melanesia. And here I'm going to show you a quick animation describing this taxon cycle hypothesis. And we start with a mainland source that's capable of colonizing this unoccupied island archipelago. And under this model, islands are colonized in a stepping stone pattern, and a new lineage evolves on each island once it's reached, shown by the phylogenetic branching on the right. Here we see a second island colonized, a third, and eventually, all of the islands in a given archipelago are filled with unique lineages. But if we wait a few thousand years, most of those lineages might actually go extinct, which creates new unoccupied island space. And this is key because it allows for this stepping stone colonization pattern to start all over again, creating a whole new taxon cycle. And this new cycle will follow the same stepping stone pattern as the first one, creating a new lineage on each island until eventually all of the available islands fill up. But because this new radiation is distantly related from the surviving lineages of the previous radiation, ecological and genetic differences between these lineages will allow them to coexist sympatrically on the same island. And this model is crucially important because it explains how islands can accumulate species and become these biodiversity hotspots, despite the fact that we don't think individual islands are big enough to support sympatric speciation in vertebrate taxa. And it's worth contrasting this model with the sympatric speciation hypothesis, which would start in the same place and follow the exact same stepping stone colonization pattern, the only difference being that in this final stage, rather than a new colonization cycle occurring, one species simply splits into two isolated lineages via sympatric speciation within a single island. And the exciting part about these two models is that despite generating the exact same patterns of biodiversity, they generate extremely divergent phylogenetic patterns. Specifically, the allopatric model predicts a distant phylogenetic relationship between sympatric taxa found on the same island, while the sympatric speciation model predicts a close sister relationship between sympatric taxa that share the same island. So, to test these models in nature, I sequenced DNA from two species of leaf warblers from Northern Melanesia, which is the island chain I've circled in red off the coast of Papua New Guinea and Australia. And these leaf warblers are distributed across the cloud forest habitat of Northern Melanesia, which I've shown here in gray. And there are just two species in the region, Phyloscopus poliocephalus, which is distributed across all of these high elevation habitats, and a second species, Phyloscopus amoenus, which is found alongside Poliocephalus, but only on the island of Columbongra. And here are some photos of the cloud forest habitat from the top of Columbongra, where these two species live and breed side by side. And P. amoenus, this single island endemic lineage on Columbongra, actually has a completely unique ecology compared to all of the other leaf warblers, where it uses its large feet and long legs to crawl around on these mossy tree trunks that you can see in the photo, while all populations of the widespread Poliocephalus lineage are canopy cleaners that eat insects from leaves near the top of the trees. And it's worth noting that the global distribution of P. amoenus is just 21 square kilometers, which is pretty incredible considering that the city of Los Angeles, where I'm giving this talk from right now, is about 50 times larger than the entire global distribution of this single island endemic species. And historically, this group has been considered a classic example of the taxon cycle, because this strange ecologically divergent species P. amoenus was assumed to be part of an ancient and now mostly extinct radiation, while the widespread P. poliocephalus was assumed to be the recent example of stepping stone colonization across this archipelago. So to answer whether this classic example of the taxon cycle hypothesis actually conforms to its phylogenetic expectations, we sequenced whole genomes from both species across Northern Melanesia. And we used an 837 million base pair alignment, essentially the entire autosomal genome, to construct a phylogenetic tree. And right away, what you see here is that we do not recover the two species as reciprocally monophyletic, and instead find a sister relationship between the two sympatric populations found in the crater rim of Columbongra Island. And if you remember our expectations, this pattern actually suggests that this canonical example of the taxon cycle did not result from the taxon cycle, but instead from sympatric speciation within a tiny oceanic island, which is a phenomenon that's nearly unheard of in birds and other vertebrates. But because of my previous experience throughout my dissertation working on taxa that displayed strongly discordant phylogenetic patterns across the genome, I wanted to search this leaf warbler dataset for any conflicting genomic patterns that might reveal more details about the group's evolutionary history. So I used ABABABA tests, which are statistical tests to identify lineages that share an unexpectedly large number of alleles. So I specified the topology we recovered from the genome-wide phylogeny, and right away you can see here this big horizontal block of significant ABABABA tests. And these 4 significant tests suggest that there's been extensive gene flow between islands within the widespread species P. poliocephalus. So this doesn't make much sense biogeographically. Why would this one tiny population be migrating across open ocean boundaries and interbreeding with other isolated high elevation populations all over the archipelago? And to explain these confusing results, I had the idea to repeat the same analysis, but this time to pre-specify the topology predicted by the taxon cycle. And with this topology pre-specified, only a single highly significant gene flow edge was required to explain the allele sharing patterns we observe. And this putative gene flow event now occurs between the two sympatric taxa on Columbongra Island, which is much more plausible because these two lineages live and breed side by side. So I then used population genetic approaches to search for gene flow across the archipelago and try to validate these ABABABA results. And in a phylogenetic network, I found that one of the P. poliocephalus samples from Columbongra Island was actually connected to the sympatric amoenus population with an additional network edge, which suggests mixed ancestry in this individual. And sure enough, when I used the program admixture, I confirmed that about 5% of the ancestry in this individual is actually attributable to the sympatric P. amoenus, providing very strong evidence for gene flow between these co-occurring taxa. And finally, I knew from the literature that avian sex chromosomes frequently resist introgression, and I wanted to see whether the Z chromosome was enriched for any specific topologies in our dataset. And when I split our data set up between SNPs found on the autosomes versus the Z chromosome, I found that the signal of admixture present on the autosomes was nowhere to be found on the Z chromosome. And when I compared topologies and sliding windows across the genome, I found that the Z chromosome is actually significantly enriched for this taxon cycle topology, where the two sympatric lineages found on Columbongra Island are distantly related to each other. And I've drawn a red arrow here on these overlaid Z chromosome gene trees where you can actually see this enriched topology that we think is an ancient history of bifurcation. So to bring this all the way back to our original hypotheses, I found that in these leaf warblers, the majority of the genome supports a sister relationship between the only sympatric lineages found in Northern Melanesia, superficially implying a sympatric mode of speciation. Yet, once we consider the evidence for gene flow recovered from the Abba tests, the more plausible explanation is that this group followed the topology predicted by the taxon cycle hypothesis, but that the sympatric lineages on Kolombongra Island experienced massive gene flow upon secondary contact. This scenario is supported by the results of our population genetic analyses and the enrichment analysis conducted on the Z chromosome, both of which suggest gene flow between these two sympatric populations. So in sum, we believe that we're observing allopatric speciation as predicted by the taxon cycle, but because of massive gene flow, a majority of the genome actually coalesces during this secondary contact event, giving the false impression of a recent sister relationship between the two sympatric lineages. And we think that P. amoenus has actually evolved such a unique phenotype and ecological specialization, not because it results from some ancient divergent radiation, but instead because of intense competition with P. poliocephalus that led to severe character displacement and forced amoenus into a completely novel ecological niche to avoid introgressive hybridization and competition for resources in the extremely restricted cloud forest habitat on the top of Kolombongra Island. So zooming all the way back out to our original question of how new species are formed on islands, we've learned that allopatric speciation is still the rule in vertebrate populations found on islands, but also that complex patterns of intra-archipelago colonization and gene flow shape the genomes of island birds. And I think that the frequency of gene flow in island systems is probably massively underappreciated because these small island population sizes speed up the dynamics of secondary contact and prevent the formation of broad hybrid zones that you might be familiar with from continental systems, where we can see ongoing hybridization in a stable evolutionary manner. Another important thing I'd like you to take away from this project is that genomes can lie. Throughout the entire field of phylogenetics, we almost always make the simplifying assumption that the dominant genome-wide topology accurately represents the branching history, but I've shown an example here in nature where this assumption directly misleads our reconstruction of evolutionary history and could have caused us to fundamentally misunderstand how new species evolve on islands were it not for the comprehensive investigation of genome-wide patterns and relatedness that we performed. And finally, I've shown that new species arise on islands via complex patterns of repeated colonization, isolation, and gene flow, and that the unique geography of island archipelagos creates an ideal arena for these forces to meet, leading to biodiversity accumulation and making island systems hotspots for global biodiversity. With that, I'd like to thank my co-authors in this project, as well as the museum collections and funding sources who made it possible. I'm happy to take any questions if there's time.
So there is time for questions. We've actually already got two of them in the Q&A. So your first one is, can you tell when the admixture occurred? Is there any evidence of adaptive introgression?
That's a great question. That's something we're working on, is using more detailed demographic modeling approaches to try to date the history of this admixture. The thing is that it's likely ongoing because we pick it up even in samples that are sampled today. So it's a pretty complex history out there.
We also have another question asking if the mitochondrial genome shows the same pattern as the Z chromosome.
That's a great question. That was one of the things that originally tipped us off to what might be going on here. The mitochondrial genome shows a similar pattern to the Z chromosome, but actually a completely unique pattern, which is like another layer of this whole thing. And we think it's a combination of resisting introgression and deep coalescence or incomplete lineage sorting because the speciation and colonization is so rapid in these groups.
All right, so at this point, we're going to switch over to our next speaker. If you have any more questions for Devon here or any of our previous speakers, again, directing you towards the Q&A where things can still get answered. In the meantime, we're going to switch to another David. So David, do you want to get your screen sharing going? go.
Okay. Well said.
And then I can just barely hear you. Can you say
Can you hear
something
me now?
again? You're still very quiet.
Oh. Can you hear me now?
Yes, that is better.
Okay, sounds good.
All right. So you can begin whenever you are ready.
Okay, great. Thanks to everyone for coming. My name is David Tian. I'm a grad student at UC Berkeley. And today I'm excited to share some of my work on understanding the consequences of small population size and isolation in the devil's hole pupfish. Oops. So species exist in nature along a continuum of small to large population size with varying levels of isolation. And when things like habitat fragmentation or landscape change occur, certain populations can become small, isolated, and endangered. And I'm particularly interested in understanding the consequences of this small population size and isolation. And so today I'm going to talk a little bit about how these processes influence genetic diversity across space and time, how they may affect fitness due to inbreeding depression, and whether they can even increase the mutation rate. But to begin, I'm going to start off by talking about my study species, which is the devil's hole pupfish, Cyprinodon diabolis. And so this is a great species for understanding these questions because they live in the smallest known habitat range for a vertebrate in the world. This is Devil's Hole, a water-filled cavern in Death Valley that measures just 3.5 meters wide by 22 meters long. In addition, thanks to biannual census counts, we know that the population size is very small. So it was relatively stable for several decades before it began to decline in the 90s. And this was punctuated by two bottlenecks in 2007 and 2013, when the population fell to as low as just 35 individuals. Following these bottlenecks, US Fish and Wildlife stepped in and created the Ash Meadows Fish Conservation Facility. And so they generated a captive breeding program, and this has been really instrumental in our understanding of the conservation of this species and has allowed for unprecedented genomic sampling access. And so to that end, I've generated 2 chromosome scale reference genomes, one for diabolis, the devil's hole pupfish, and another one for Neanderthals, a closely related desert pupfish. And in terms of continuity, accuracy, and completeness, these are just really high quality reference genomes that we're working with. In addition, we've sampled genomes from all 9 species within Death Valley. And so what you see here in the middle is a depiction of what Death Valley would have looked like about 10,000 years ago. So it's covered by these large pluvial lakes where the pupfish intermingled. But obviously since then, as the environment has become more arid, these pupfish have become isolated in these independent springs and ponds. And so here I'm just showing the sampling locations as well as the variety of habitats in which these pupfish live. And so to get at the first question that I outlined earlier, how does small population size and isolation influence genetic diversity? So first, I just want to go over a little bit of the demographic history of the system. So using MSMC, which looks at the distribution and density of sites in the genome where the haplotypes differ, we can infer historic effective population size. And what we see is that for many of these species, population sizes began to decline about 10 to 20,000 years ago, which coincides with when Death Valley began to dry up. In red, we have the devil's hole pupfish, and we're not able to reconstruct the historical effective population size, but through simulations, I've shown that this is a result of extremely low genetic diversity, causing MSMC to not work properly. In addition, I just want to note that there's an extensive history of gene flow. So using genetic data, specifically F statistics, which look at the variances and covariances in allele frequencies between populations, we're able to model demographic history. And so there's a lot going on here, but the main takeaway that I want you to take away is just that there's a lot of these dotted edges that represent admixture. So historically, there's just a ton of gene flow and admixture throughout this Death Valley pupfish system. But today, all of the water has dried up, and so these pupfish are isolated, and there's really strong population structure. So we can see in PC1 separation of the Death Valley pupfishes from other outgroups in Arizona and Mexico. And we can also see separation of devil's hole in red from the other Nevadensis and Salinus species in Death Valley. And these results are more or less recapitulated within our admixture results. So for the most part, for all these different species, we see that most of these bars are solid colors. There's not that many bars with mixed colors representing mixed ancestry. In terms of genetic diversity, we measured genetic diversity in terms of pi. And so what we see is that across the board, there's very little genetic diversity within these Death Valley pupfishes. But within Cyprinodon diabolis, the double-soled pupfish, it's up to an order of magnitude lower. Its genetic diversity, just to put it into context, is somewhere in between that of the vaquita and one of the Channel Island foxes. And so I also just want to note that the captive population is actually even less genetically diverse. And this is despite the fact that the captive population, in terms of the census size, is much larger at about 500. And so we think that's indicative of a large amount of reproductive skew within the captive breeding program. But how has genetic diversity changed over time? So to get at this question, we looked at, we sequenced genomes from historical specimens that were formalin fixed with the UC Santa Cruz Paleogenomics Lab. And so we were able to sequence genomes from as early as 1941. What we found is that despite recent population decline, there actually hasn't been that much reduced genetic diversity. And we think that's because genetic diversity in the past was already extremely low. And so even if we expect relatively large relative differences in genetic diversity due to population decline, that might still result in low absolute differences between the historic and the modern if the historic was already very low. So to answer the first section, small population size and isolation have led to really low genetic diversity across space and time within this Death Valley pupfish system. And now I'm going to move on to some work that I've done to study its effects on fitness. So about 2 years ago, U.S. Fish and Wildlife notified us that about 25% of the embryos growing up in the captive refuge program were dying at about 5 days post-fertilization, and some of them had a heart defect. So on the left, we have a wild-type individual. You can see that its heart is beating vigorously. This individual will grow up into a mature adult. But on the right, we have this sort of heart defect individual. At 5 DPF, it's going to die very shortly. We can see that the heart is not beating very strongly. There's enlarged melanocytes. In general, it's just not very healthy at all. And so when we compare heart rates, we can see that the embryonic lethal individuals tend to have a much lower heart rate, about 23% lower than that of the mature adults. And so we sort of wondered whether this was indicative of inbreeding depression within our system. And so to do that, we first started off by measuring individual levels of inbreeding. And we did that with this metric known as FROH, which we defined as the percent of the genome made up of these runs of homozygosity that are at least 100 kb long. And so this is what we found. So in the plot on the right, I have the length of the genome that is made up of runs of homozygosity along the x-axis. Along the y-axis, I have different individuals. And what we find is that captive individuals tend to be much more inbred than the wild ones, which lines up nicely with our genetic diversity results. We also find that the embryonic lethal individuals tend to be much more inbred than the mature adults, which potentially signifies inbreeding depression. And I've also colored the bars in terms of the size distributions of the runs of homozygosity. And so you can see that for many of these embryonic lethal individuals, they have a lot more of this dark red maroon color, indicative of these larger runs of homozygosity that are expected to have arisen relatively recently before recombination can break them down into smaller sizes. And I also just want to note that we were able to sequence one of our museum specimens from 1941 up to at least 15x. So we've included it here, and it suggests that the population was relatively inbred even back then, consistent with our temporal genetic diversity results. But how much does this difference in inbreeding actually affect fitness? And so we made a generalized linear model with a binomial distribution, and we found that just a 10% increase in inbreeding is associated with a 75% reduction in the odds of survival. And so this is indicative of really strong inbreeding depression within our system. And I think it also suggests that we need to be studying inbreeding depression in the wild, not just at the adult stages, but across other life stages where inbreeding depression may vary. And we also wanted to take a look at the genetic architecture of this inbreeding depression. So what you see here is a GWAS, where we're just looking at this binary trait of are you alive or are you dead? And in terms of the genetic architecture, it's relatively oligo or polygenic. There's a lot of peaks dispersed across the genome, and some of these hits coincide with really crucial genes involved in maintaining homeostasis. So things like ribosomal proteins, translation machinery, heat shock proteins, and we're still digging through this list of candidate peaks to try to figure out what's going on exactly. And one of the ways in which we're doing that is through also doing some homozygosity mapping. So there's this one region along chromosome 16 where there's a 2 megabase region where the vast majority of embryonic lethals have a run of homozygosity, and most mature adults do not have a run of homozygosity. And so this is a peak that we're looking into more deeply, so stay tuned for that. Okay, so to conclude that second section, we find that small population and size and isolation have driven some pretty serious fitness, effects through this inbreeding depression. And I'm going to wrap this up by talking a little bit about my work to estimate a mutation rate that might be a function of this small population size. So germline mutations are the source of all heritable variation. And as evolutionary biologists, we're interested in understanding the rate at which these mutations occur because they provide direct relevance for things such as estimating how quickly populations may adapt, as well as estimating population divergence times. And across vertebrates, there's over 40-fold variation in these mutation rates. And there's a couple of reasons why we think the devil's hole pupfish might have an elevated mutation rate. And one of those reasons is the drift barrier hypothesis. And so this hypothesis predicts the evolution of higher mutation rates in species with long-term small effective population size. And the logic behind this is that generally we think of mutations as being deleterious. And so in small populations where there's reduced efficacy of selection, potentially you could have a case where you have a mutation in a DNA repair protein and selection is unable to purge it from the population because drift overpowers selection. And so there's evidence across vertebrates, but also more broadly across eukaryotes and prokaryotes that suggests this negative relationship between population size and mutation rate. And so normally we would calculate a mutation rate with a pedigree, but I don't have one. And we did all of our sampling at 1 generation, so we were not able to statistically infer parent-offspring relationships. And so I used a different method that's been used in human populations with consanguineous marriages. And I'm happy to talk about it more in the Q&A later, but very briefly, it relies on inbred populations and identifying identical by descent segments that are autozygous, finding heterozygous SNPs within these autozygous segments, and then leveraging that to calculate a mutation rate while taking into account the length of the genome over which we're searching for these putative de novo mutations, as well as the number of separating meioses, so taking into account the amount of time that may have passed as we're calculating this mutation rate. And so when I do that and take that approach, we calculate the devil's hole pupfish mutation rate to be about 3 × 10⁻⁸. So you can see on the plot on the right, there's quite a bit of variance in these mutation rate estimates, largely due to uncertainty surrounding the number of separating meioses. But we're basing this rate on our 4 super high coverage individuals, the 4 red ones. And so if we take that average, it's about 3 × 10⁻⁸. So this rate of 3 × 10⁻⁸ is about 5 times higher than the average rate for fishes and N of 1, but it does support the drift barrier hypothesis. And I think in the future, there's really interesting opportunities to study mutation rates for other populations that have been at long-term small population size to get further data on this relationship. So to conclude, I just have some major takeaways. So among these Death Valley pupfishes, there's a lot of historic connectivity with historically larger population sizes. But as the environment changed, we got a lot of strong isolation as well as population structure, which led to really low genetic diversity across the board, but particularly so for the devil's hole pupfish. And in this system, it's remained relatively consistent across the last century. We also see evidence of really strong inbreeding depression. So just a 10% increase in inbreeding is associated with a 75% reduction in survival odds. And we also find an elevated mutation rate that happens to be consistent with the drift barrier hypothesis. So with that, I want to thank my lab, our collaborators, the natural history museums, funding sources, and if there's time, I'll take some questions.
I'm sorry, but with like 30 seconds until our next speaker, I don't think we're going to have time for live questions. We do have the Q&A. We also only have 5 speakers, so we can potentially do additional questions after the 5th speaker. But for now, Matthew, could we get you to share your screen?
All right, yes.
Right. And you should feel free to begin whenever you are ready.
All right, thank you very much. Hello, everyone. I'm Matthew Treaster. I'm a graduate student in Mike White's lab at the University of Georgia. And today I'll be talking about sex determination in threespine stickleback fish. I want to begin by defining this term sex determination. While many animals have 2 separate sexes as adults, early in development, the embryo has no sex and exists in this bipotential state where it has the capacity to become either a male or a female. Sex determination is this really precise decision-making point that begins the downstream differentiation of the embryo into one of these 2 sexes. While this downstream differentiation network is well conserved across many species, this initial cue of sex determination is surprisingly varied, and it can be regulated by a wide range of mechanisms, including environmental factors, social cues, and genetics. We'll often discuss primary sex determination, which governs the differentiation of the early gonad into either a testis or an ovary. And in genetic sex determination, which I'll be focusing on today, this is regulated by a single sex determination gene, often found on a sex chromosome. For example, this Y-linked sex determination gene would initiate development of a testis in the XY genotype, and in the absence of this gene, ovaries would instead develop in the XX genotype. Genetic sex determination has evolved countless times across the tree of life, and our lab is really interested in understanding how a new sex determination gene evolves. We study this process in the threespine stickleback, which have an XY system like the one found in mammals, with XX females and XY males. When our lab assembled the Y chromosome, we found this candidate sex determination gene, AMHY. This is an additional copy of the gene AMH that was duplicated onto the Y chromosome around 22 million years ago when it first formed. So, why do we think AMHY is the sex determination gene? AMH, or anti-Mullerian hormone, is an important gene in male differentiation across vertebrates, so we know that it has ancestral function in gonad development. More importantly, independently derived Y-linked copies of AMH have been found in at least 10 different taxa and have been shown to have function in sex determination in several groups. But despite its widespread convergence as a sex determination gene, we still don't have a great understanding of how AMH regulates development of the undifferentiated gonad, or how it evolves this novel function, moving from a downstream differentiation gene that responds to sex determination into an upstream regulatory factor. But before we can address these big picture questions, we first have to figure out if AMHY is the sex determination gene in threespine stickleback. To answer this, we've used CRISPR-Cas9 to knock out AMHY in XY fish and introduce AMHY into XX fish using a transgene that contains AMHY and its surrounding putative regulatory elements off of the Y chromosome. If AMHY is the sex determination gene, we predict that we would see sex reversal where XY fish without AMHY develop ovaries and XX fish with AMHY instead develop testes. We screened for sex reversal in AMHY mutants using histology of the gonads. Here I'm showing a wild-type XX ovary and XY testis. Ovaries are easily identified by the large oocytes, the female gametes. They start as these smaller primary oocytes that grow substantially in size as they mature. In contrast, on the bottom we have a testis. All of the dark spots that you see are the complementary cells, the spermatocytes, the male gametes, which are then separated into these large compartments or cysts. So what about the AMHY mutants? In XX fish with the AMHY transgene, we see female to male sex reversal, and these fish develop clear testes just like those found in wild-type males. And XY fish without AMHY show male to female sex reversal and develop ovaries that even have mature egg cells. We were super excited to see these phenotypes, and we're now confident that AMHY is the sex determination gene for our species. However, we were a bit surprised at just how, well, normal these sex reverse gonads looked. In mammals, mismatches between the sex chromosome genotype and phenotypic sex often results in poor gonad development and a reduced ability to make viable gametes. But since we didn't see any signs of aberrant development, we wanted to assess if the gonads of sex-reversed fish were functional and measure the fertility of the AMHY mutants. When we compare the wild-type XX and mutant XY females, we see that although XY females produce a similar number of eggs per clutch, these eggs show dramatically reduced viability. We also find that XY females are frequently egg-bound, which you can see in this individual here, whose abdomen is much more distended and irregularly shaped than a wild-type gravid female. While gravid females can typically pass their eggs once they're mature to begin another round of meiosis, in these egg-bound females, the eggs get stuck, quickly lose viability, and can even prevent the fish from producing the next clutch of eggs. We think that the impaired fertility in XY females is due to this tendency to become egg-bound rather than an inability to produce viable eggs, and we found that by manually removing the eggs from the fish, we can get some crosses with viability similar to wild type. In these crosses, the XY females can transmit either the X or the Y chromosome, and they do so with equal frequency, so we don't see any signs of segregation distortion. However, we have found that YY embryos are not viable, likely due to the many genes that have been lost by the Y chromosome. As for XX males, they are fully fertile in our ex vivo laboratory crosses. This makes sense as the histology shows the males make mature sperm. You can see tiny condensed dark nuclei of the sperm heads and the long sperm tails in this histology slide. But it does make us wonder, what the heck is the point of the Y chromosome if you can be a fully fertile male with just this single gene AMHY? So to think about the function of sex chromosomes, it's helpful to consider how they form. Sex chromosomes originate as a normal pair of autosomes. One of these autosomes then acquires a sex determination gene, such as AMHY, and the leading model predicts that if there is a nearby locus that is beneficial to one sex, but neutral or even detrimental to the other, suppression of recombination may be selected for to link this allele with its favorable sex. Over time, this process can repeat and continue, and the sequence in these non-combining regions will diverge from one another to create the striking heteromorphic sex chromosomes that can be found in many species. So what sorts of male beneficial alleles might we find on the stickleback Y? The stickleback Y chromosome has undergone significant evolution since it acquired AMHY. It's lost around half of the 1,200 genes found on the ancestral autosome that it evolved from. It's also acquired a number of Y-specific genes that are not found on the X. We know from previous work in our lab that many of these Y-specific genes are upregulated during spermatogenesis, so we were surprised to see that they're not actually necessary for this process. There are other avenues through which we predict the X chromosome could be impacting fitness. Sex chromosome genes may affect things like sperm motility that could play a role in sperm competition, which is common in externally fertilizing fish like the stickleback. These genes could affect many of the unique behaviors involved in reproduction and paternal care that sticklebacks are well known for. They also could be impacting mating displays that help a male stickleback attract their mate. Stickleback's best known mating display is this blue and orange nuptial coloration that can be seen in males during breeding seasons. This coloration serves as an indicator of fitness to prospective females and helps them choose which male to mate with. Now, this coloration is not as vibrant in the lab, but it's still clearly visible in wild-type males. All fish begin in this plain silver color, like the first wild-type female shown here, and females may darken slightly when they become gravid, but they're still quite distinct from the males with these dark blue eyes and bodies. We find that XX males do not fully recapitulate the nuptial coloration of wild-type males, but they do show noticeable blue pigmentation on their bodies and eyes that we don't see in XX females. But what I found even more exciting was that XY females also show elements of male nuptial coloration, which, though more variable, is present in both the eyes and bodies of multiple individuals shown here. These findings show that primary sex determination, testis differentiation, and the sex hormones that they produce alone is not sufficient for male nuptial coloration, and that there are likely other X or Y-linked factors involved in this important reproductive trait. So to wrap up, we've shown that AMHY is the sex determination gene in threespine stickleback, which establishes the species as a great model to investigate how this gene has repeatedly evolved this function across multiple species. We've shown that the Y chromosome is unnecessary for male development and sperm production, but this doesn't mean that it has no purpose, as we also show that sex chromosomes can contribute to secondary sex characteristics and thus fitness in ways that are completely independent of their role in sex determination. Stickleback feature many sexually dimorphic traits and well-studied reproductive behaviors, and this new ability to separate gonadal sex from sex chromosome genotype provides an exciting new avenue to study the genetic basis of sexual dimorphism. And finally, while I've spoken a lot about gonads and the single gene AMHY, sticklebacks show us that sex is multifaceted and can't be easily reduced to just a single genotype or what gametes are produced. I want to quickly thank the members of my lab and my committee, all of my sources of funding, including the Society for the Study of Evolution, the conference organizers for giving me the honor of presenting to you all today, and then last but not least, all of the fish. And with that, I am happy to take any questions.
And we have plenty of time for questions because you finished in 10 minutes. So, as always, feel free to either raise your hand or post something into the Q&A. All right, let's ask our first person to unmute. There we go.
Hi, great talk. I was just wondering if you have looked at the comparative this sex determination gene with some other sex determinant gene like SRY gene in Drosophila. So have you tried that and have you found any similarity in them?
There's been a lot of comparative work into different sex determination genes. And I'm mostly familiar with those invertebrates. And what's really interesting is that you see a really wide range of variety where you have genes like SRY or DMRT that are transcription factors, and we can see, that they directly initiate differentiation of somatic cells in the gonad. We see some enzymes that are involved in hormone production. But then we also have AMH, which is the most prevalent factor. It's the most converged upon sex determination gene. That's a secreted hormone. And it's less clear about what exactly it's doing in development of the gonad. There's not any apparent similarity to some of the other genes like SRY or these other transcription factors, but we do also see other similar hormones to AMH or even repeated evolution of AMH acting in sex determination.
All right, we've also got, we just got one here in the Q&A. So are you planning to run any behavioral tests on these genetically modified individuals to test for phenotypic changes in other nuptial behaviors?
That is definitely something that we're interested in. I don't know if our lab specifically will do this. There's also, a lot of other stickleback labs that specialize in behavior. So we're, talking about the possibility of us working on that or collaborating with them. But yeah, there's so many sexually dimorphic behaviors and so many well-studied nuptial behaviors that the males exhibit, and both in like nest building and attracting a female, but then also in that paternal care that is pretty unique with stickleback. And yeah, we're really interested to see if the sex chromosomes play a role in these traits, which has been seen in mammals, that the sex chromosomes do affect paternal care.
All right, Catherine, I saw that you had your hand up.
I had the same question.
All right.
Okay.
So Earl has asked in the chat, actually, if you looked at sperm motility in XX males.
We have not yet, no. We did just these really basic fertilization assays. So we macerate the testis and apply the sperm solution directly to eggs. So, it doesn't require much motility for them. But we are interested in looking at, just sperm morphology and motility to see even if the sex reverse males have functional sperm in the lab, perhaps they wouldn't be as functional in the wild. And also doing sperm competition assays to see if even if a male could mate in the wild, could they be outcompeted by wild type males due to these other factors on the Y chromosome?
Right, so that is our set of the Hamilton speakers. I am not envious of the judges having to make decisions about any of them. I will point out that since we only had 5 speakers in this session, we do have time if anyone wanted to ask any other questions of any of our speakers in this session, I will change the permission so that you're all actually able to unmute yourselves if you need to.
While people think about if they want to ask questions, I just want to thank all the Hamilton finalists for your outstanding talks. That was so much fun, so much cool science being done, really outstanding work. So thank you all. Yeah, this is not going to be easy, but you should all be very proud.
All right, if there are no more talks at this point, we will wrap up this session. So that is the end of our first day of talks. We will be reconvening for another day of talks. If you are in the New World, it'll be tomorrow. If you are in Europe or Asia, it will be much later on today, as people will have a chance to potentially get something to sleep. So go grab something to eat, get away from your computers for a little while, and we will see you back here somewhere in the neighborhood of 13 hours from now.